And I know there was his presentation has a title, and you have that in your program, you have that on the slides. I think the subtitle of his presentation was probably "Please Advance." Well, it was good. It was excellent information. And next, we're going to go a little deeper on the Powassan virus. The tick-borne virus has great human health concern now across the state of New York. Maybe this will be the end of, please advance with this little thing. Yeah. Yeah, Dr. Saravanan Thangamani is already on his feet. He knows more than a little bit about ticks because he has collected enough of his own to start a museum, and he's done tremendous work across the state of New York and in several other states to help track, identify ticks, and study what diseases they carry. He's one of the leading experts you would find anywhere in this field. Ladies and gentlemen, Dr. Saravanan Thangamani from Upstate Medical University.
All right, good morning, everyone. So I'm going to kind of cover a topic that I think not very well discussed in any tick meetings to go. It's one of the neglected, often under-investigated pathogen amongst the tick-borne disease field. Somehow I got hooked into this 15 years back because I had nothing else to do. And I always believed in road not taken. So I decided to take something that no one else is investigating. So 15 years back, I focused on Powassan virus, and still the journey continues. But the first slide I want to say is that, and often we think that deer ticks are at the tip of the blade, the grass blades, but however, ticks can be everywhere, right? So I'm not sure if I can actually play the video. Yeah, if you can tell, zoom your eyes on where the ticks are. Even the dirt path can actually have ticks. It's not just by the edges, not by the grasses, even by the dirt path you can have. So I just want to highlight that. So my outline is going to be, I'll briefly give an introduction. It's going to be really basic introduction because it's not like a Lyme disease. Everybody in this room is very well aware of it. I'm going to talk a little bit about ecology of the Powassan virus transmission, and then what kind of clinical symptoms one would actually see in humans, and then how it has been diagnosed, and a little bit about epidemiology and geographic expansion. And I will finish it up with a few slides on what my lab has been doing. And my lab has a larger portfolio, but I'm going to focus on two key that is unpublished that I'm really excited about. The lab is taking the direction. So with that, so Powassan virus is a flavivirus similar to Zika virus. However, it's transmitted by deer ticks. Was first isolated in 1958 from a young boy who died of encephalitis in Powassan, Ontario. That's like 6 hours north to us. That's how it's called the name. It's got the name Powassan, and it's closely related to another flavivirus, Tick-Borne flavivirus, circulating in Europe, Russia, and China and Japan, where nearly 50 to 60,000 human cases are being reported each year. And the primary vector at that time was identified as Ixodes cookae, otherwise called as groundhog ticks. In 1996, researchers from Harvard, they actually identified another tick-borne encephalitis-like virus, but this time it was from a deer tick, so they called this deer tick virus. The reason being is that when they did a genetic analysis on this virus, it was not 100% homologous to the Powassan virus that was identified in Canada. However, it was about 96% homology, and they did a phylogenetic analysis here, and it's clear that it is indeed a different virus. So instead of calling a different virus, they call it a lineage 2. So the virus that was originally discovered in Powassan, Canada, is a lineage 1 virus. The virus that was discovered in Connecticut and Wisconsin at the time was called lineage 2 Powassan virus. So how is it being transmitted? I think Brian kind of walked through this very nicely. So what I can actually focus is something that, for very specific to this virus is the adult ticks. Sorry, you got to kind of, my pointer is not working. So you got to focus on the adult. When the adult feed on larger mammals, they lay thousands of eggs, and then that becomes larvae. When a larvae then finds a susceptible small mammal, it then feeds and then molts to a nymphal stage tick, and then nymphal stage tick then finds medium-sized mammals and then go to few weeks after that, it will become either male or female tick, right? So that cycle goes in. But however, if that small mammal is carrying the virus, when this nymph larval tick attached to them, acquire the blood, now the tick will acquire the virus as well. The same thing happens for the nymph, and also same thing happens for the adult. Unlike Borrelia burgdorferi, that is not transovarily transmitted, meaning that from the adult to the eggs, viruses can be transmitted from an adult to egg. So imagine a single female positive tick can actually lead to several thousand positive larvae, right? That is hypothetical situation that we have proved in the lab. How they are perpetuating in nature is the biggest question. We still don't know how the virus is perpetuating in nature. We know that they will acquire from a small mammal to the tick, and then it can actually, once the tick is positive, it will be positive for his entire life. There's another differentiation between the Lyme disease agent and this. So Powassan virus, we know that the primary vector is Ixodes cookeyi. As the name suggests, it's a groundhog tick. Its entire life cycle, natural history happens in and around groundhog. We don't know what other species that it commonly feeds on. And for the deer ticks, we all know that it feeds on pretty much a variety of small mammals, medium-sized mammals, and larger mammals. But how virus is perpetuating within itself is that there are multiple ways they can perpetuate, which means that an infected tick and an uninfected tick can feed very close to each other. They can actually form tunnels. And that's one way they can actually transmit the virus. The other one is from the mom to the babies, from the adult tick to the larval tick, they can transmit. And transstadial transmission means from larvae to nymph. So one life cycle stage, another life cycle stage, that's called transstadial transmission. Venereal transmission means that if a male tick is positive and the female tick is negative, when they mate, they transmit as well. So this is in a way sexually transmitted disease as well. And the other one is through the reservoir. So we know that deer ticks feed on multiple different hosts, but we still don't know what is the primary host that actually perpetuates the virus. We don't know that yet. So that is a lot of questions that we have to answer that. So that is kind of an interesting thing. And you will know why I'm actually spending more time on the reservoir in one of my latest other slides. In addition to that, so we now know that groundhog ticks and deer ticks are the primary vectors for these two viruses, but recent research has demonstrated that dog ticks and Lone Star ticks that are slowly emerging in the New York can actually transmit Powassan virus as well, can acquire and also transmit as well. And in our own lab, we have collected Powassan virus lineage 1 from a field collected dog tick, which means that in addition to laboratory research is proving in the lab, we are also finding it in the lab field as well. So this could be the future, health concern or human health concern that additional ticks will now be competent to transmit Powassan virus and deer tick virus. So coming back to the clinical manifestation, if an infected tick bites a human, within the first approximately a week, the first sign of symptoms one would notice is febrile illness, like any other illness, fever, sore throat, drowsiness, headache, and disorientation. And if they have very strong immune system, if the virus is not a virulent virus, it will then actually go to the encephalitic disease phase, which is, loss of coordination, speech difficulties, paralysis, and seizures. If they are lucky, they will go to the neurological sequelae stage, which is all your life, you will have the neurological sequelae. If you're unlucky, you'll be one of those 15% cases that you'll succumb to the disease. So it can cause fatal neuroinvasive disease in 10 to 15% of the cases. Long-lasting neurological sequelae have been documented in 50% of the survivors. So that stays with you for your entire life. And some infection may only cause febrile illness or asymptomatic. So the initial first 1 week is mostly asymptomatic. If someone goes to the febrile illness, I don't think that any of you will actually look for Powassan virus diagnosis. I'm certain about it, right? You will not see this. So those are the undocumented cases that easily got misdiagnosed or undiagnosed, I would call it. So here is a classical example. I like to show how the virus actually from initial infection, it can actually cause significant disease within a matter of few weeks. So this is an MRI image from a fatal case of Powassan encephalitis on admission on panel A, and after on B is 8 days posted hospital admission showing severe inflammation in the cerebellum and the midbrain region. So the patient lost consciousness and capacity for independent respiration. 2 days after admission and died on day 13. So it's pretty fast resolving disease, not resolve, fast, disease that actually, once it gets into the brain, once it crosses the meninges, you certainly have encephalitis. And if your immune system is not good enough, you will succumb to the disease. So generally speaking, Powassan encephalitis cases are typically characterized by perivascular and focal parenchymal infiltration and monocytes. You would see occasional brain necrosis associated with areas of more intense inflammatory infiltrates. Widespread destruction of neuronal cells have also been reported in the large motor neurons of the brainstem and spinal anterior horns, cerebellum, basal ganglia as well. So how are we diagnosing this? So this is the biggest mystery as well. I have to tell you that, because if some patient comes to your clinic with an acute febrile illness, you will certainly not diagnose Powassan. If someone has a neurological consequence or meningitis, you will certainly not go check for Powassan. When everything else, you can't diagnose anything else. And then if the patient say, I was exposed to a tick bite, and that's when the light bulb will go, okay, I should look for arboviral encephalitis, particularly Tick-Borne, and that's when you will do this. So there are multiple ways one actually can diagnose: from the blood, from the cerebral spinal fluid, from the brain tissue, but obviously the brain tissue is from a terminal patient, right? So blood can be used for virus isolation. You can use PCR, like COVID test. You can use sequencing, and also you can use serology, detect IgM and IgGs. And CSF can be used for virus isolation, PCR, sequencing, serology as well. As far as the CDC guidelines go, a positive Powassan IgM in the CSF followed by a PRNT, or neutralization assay, or negative IgM for any other arboviruses that are circulating in this area. In our area is West Nile virus, Eastern equine encephalitis. So that is the criteria that CDC has. And I don't think any clinic other than NYDOH can actually do a confirmatory test on Powassan samples or for diagnosis. So you send the sample to DOH, they then keep half of the sample, they do their own test. And then if they find it positive, they then send the other half to CDC, and CDC does the confirmatory testing. And then the message is relayed to the DOH, and DOH then relates back to the clinic. So by the time the patient probably lost consciousness, probably dies. So that's the situation we have now. So that's a worst case scenario. I'm trying to exaggerate here. So the reason why I'm exaggerating is this. So since 2004, between 2004 and 2022, we have 288 cases. It pales in comparison with SARS-CoV-2, right? But that's why I said the road not taken. I like to work on things where nobody works on it. And amongst that, almost 99% of them are neuroinvasive cases, because you go to the clinic only when you have neuroinvasive disease. If you have acute febrile illness, you'll stay home. You'll take Tylenol, stay home, right? So these are the cases that where people actually went to a clinic, the doctor actually diagnosed it. And of that, 35% of unlucky people succumb to the disease. And you can see from this map here, pretty much all the Powassan cases are concentrated in the northeastern United States and the Greater Lake region, right? And New York is that epicenter of that. So if you look into the age and the sex, you can tell that kids under 18 and older people about 60 are the primary population demographic that actually gets the disease. But if you kind of dwell deep into the male and female ratio, you will see there is some sort of disproportion between males and females. And I attribute that to males don't follow the instructions we are giving them to do the tick check, but females follow that. So that is one reason. I certainly, when I buy a coffee maker, I don't read how I need to install it. I just go and assemble it, disassemble it, right? My wife reads it, the entire manual how to do it. So that's why I tell males don't read the instruction or don't hear the instructions. So what time period of the year where most of the Powassan cases are diagnosed? As you can see here, summer and the fall. Those are the predominant times where Powassan cases have been reported. However, you can see that throughout from January to December, there's always been a case. So, oftentimes people used to think that there is a tick season. Actually, there is no tick season. Every month is a tick season in a way. You can imagine that a disease that is as low incidence as Powassan, whereas can be detected, diagnosed in January and December, or in January and February, the peak is winter months. You can detect Lyme disease, you can get anything, right? So in the year 2023, so we are halfway through 2023, there's already 24 cases have been reported, and you can see that New York again is epicenter. So we do see a lot of cases here. Of that, all of the cases that have been reported to CDC are neuroinvasive disease cases. As I said, you don't go to the clinic if you don't have a neuroinvasive disease. That's period, right? So just want to kind of give an idea. The original discovery was done in 1958. And then over the years, every decade, slowly the number of our human reported cases have increased. So what we believe is happening is that, wow. So what is happening, what I think is happening is a geographic expansion of the Powassan virus in the United States. So in America, although the original virus discovery was done in 1958, in the United States, the very first human case was in 1970 in New Jersey from a human patient. From then on, with the next few decades, Powassan virus kind of expanded all the way throughout the New England and Greater Lakes region. Now we are seeing the Powassan virus actually starting to appear in southern states as well. So what we think is happening is that on the bottom left, where you see is the map of the distribution of deer ticks in the United States. And then if I overlay that on the Powassan virus, and you can clearly see that wherever deer ticks are there, Powassan virus is also emerging. So what we think is going to happen in the next decade or so is that more and more Powassan cases will be appearing in the southern part of the United States, including Florida and Texas as well. So that's what we think is happening. That's our crystal ball. And what do we have for vaccines and countermeasures? Zero. We don't have any vaccines or countermeasures because nobody wants to develop for a disease that only infects 288 people in about 20 years, right? So there is no economic incentive for any pharmaceutical company to invest in it. However, as researchers, we like to do our own investigations. So we did, the first one, the first novel synthetic DNA vaccine that I showed there was part of our lab as well. And then Moderna, even before the COVID vaccine was actually made popular, they actually investigated in Powassan virus to develop an mRNA vaccine. It's the same platform that we are getting the vaccine. And there are other people that used therapeutics, monoclonal antibodies to see if they could use that as a way to prevent or limit the spread of Powassan virus. However, it all works in a laboratory setting. As you can see from here, 100% protection was achieved in a laboratory setting on a small animal model, but so far, no one is actually taking it beyond that. So it is more of an academic interest right now. So there is no specific FDA-approved vaccine or therapy. For Powassan cases requiring hospitalization, supportive care is provided, and it often involves respiratory support, intravenous fluid, in the aim of reducing the cerebral edema. So mostly you will have meningitis and encephalitis. And minimizing tick exposure is the best and the only way to prevent getting a Powassan virus infection and also any other tick-borne disease infection. But I must admit here is that for the rest of the tick-borne agents, if you pick the tick, maybe a day after that, you will be okay. But this virus is not like that. The virus is delivered to the human skin immediately upon tick attachment. As soon as the ticks are actually depositing this cement protein, it is depositing the virus also. So which means that in this case, you got to prevent not getting a tick bite. Not doing tick checks is not going to work here. You will have virus in your body. But will you get sick with that virus? It's a big question. And you have an answer at the end of my slide. So that brings back to some of the key things that lab is doing. So my lab has been working with the Powassan virus for nearly 15 years. Since I started my independent lab, my primary lab when I started was focusing on the feeding site of the tick. When tick is feeding, it delivers virus at the skin level. So my lab focused on understanding how the tick is actually delivering the virus to the human skin. So if we can understand how the virus is delivered, we can actually develop novel countermeasures to block the transmission. So the lab was designed in a way to develop transmission blocking vaccine laboratory, but then we kind of spread across our wings differently. And some of the key publication came out of the lab based on that is that. And after moving to Syracuse, I started a new portfolio, which is the ecology of Powassan virus transmission. And that is the reason why we become a little bit more popular, because you will see why. So basically what we try to do is to actually do a One Health holistic approach surveillance to understand why the virus is not perpetuating Because if a female is positive and it can lay eggs, thousands of eggs, and if those thousands of larvae are positive, why we don't see more human cases? What is the reason? And also Powassan virus infections are highly focal, only in the lower Hudson Valley. We don't see it in Onondaga County. Why? We don't know that. Deer ticks are everywhere, but it's very highly focal. So we were more interested in understanding what determines the focality, and what are the factors that determines the Powassan transmission, perpetuation, and also we don't know what the reservoir is. So that's a primary question we started that. It is an ongoing work we do. And on the right, you see key contributors to the Powassan virus research in my lab. So before I show the exciting one, I want to kind of show you some boring ones, which I kind of earlier alluded that Powassan virus, deer tick virus, one is transmitted by groundhog tick, the other one is transmitted the deer tick, but we kind of put everything under one basket Powassan virus. It's not true. You can call it Powassan encephalitis, that's perfectly fine, but you can't call that as a single virus. That's my pet peeve. And we all know from genetic analysis, these are two different viruses. You know, there's only like 89% genetic similarity between us and the chimps. There's 98% homology, right? And this is 89%. So which must be, they must be doing different things. So our lab was more interested in actually seeing, okay, what is really happening between these two viruses? Do they cause similar disease? Do they cause the same neuroinvasion into the brain? Do they infect the same neuronal cells in the brain? So we started asking a lot of questions. So what I'm going to show here is very preliminary. So we did a mouse model. So unfortunately, we can't do it on humans. So we got to do on small mammals. So we inoculated both the viruses at the same time, same dose, everything at the same time, same batch of mice. What we ended up seeing is that all the mice, whether they received deer tick virus or Powassan, they all succumbed to the disease within 10 and 12 days. So we were like, okay, that's a dead end for us. But then I started to see the clinical scores of how the mice succumb to the disease. Because we normally have a clinical chart of, we measure 16 to 17 different parameters every single day, how the mouse is behaving. When we looked into that, when we put a scoring chart to it, we saw a difference. Deer tick virus succumbed to the disease primarily because of weight loss and seizure. Powassan virus, on the other hand, succumbed to the disease because of paralysis and paresis. So that was a light bulb moment for us. Okay, something is happening. They all succumb to the disease, true, but how they succumb to the disease is different. So that was our scientific question. We wanted to investigate, okay, what determines the clinical outcome? So we then actually took all the internal organs of the mice. We actually looked for the viral titer. As you can see from here on the left part of the brain, the green ones from now on, it's basically deer tick virus. The purple ones are Powassan virus. And we saw a tremendous difference in the viral titer between Powassan virus and deer tick virus. And they're all different organs, brain being the most significant because obviously this is an encephalitic virus. So that led us to believe that definitely there is something else is happening. So we decided to invest our energy and time into doing additional stuff. I'm not sure. I don't see it clearly here. All right. Whenever you see a teal color, that means that is a virus. So you can see on the left is a deer tick virus. On the right, the brain section that you see is a Powassan virus. And you can see a differential tropism already there. Deer tick virus primarily like to feed on cerebral cortex, hippocampus, and thalamus. Powassan virus, on the other hand, likes to infect the cerebellum primarily, cerebellum brainstem primarily. So the green ones on the schematic diagram I show, those are the statistically significant change between Powassan and deer tick virus. So we now can significantly conclude that Powassan virus prefers cerebellum and deer tick virus prefers cerebral cortex, hippocampus, and thalamus. That actually correlates to seizure, like infection of Powassan virus neurons in the hippocampus in a way leads to the seizure. So that correlates. Our histopathology data now correlates with our clinical observation. And the infection of Powassan virus in cerebellum, brainstem, and the spinal cord, that leads to the paralysis. So we now have a clear evidence that these two viruses actually have two different modes of neuroinvasion, cause differential clinical outcome. However, this is a mouse model. Mouse is not humans. So we are hoping to reach out to a lot of clinicians who actually have access to the patient who succumbed to the disease, so we can actually look into the brain sections to see, are we seeing a similar pattern here? So that is something that we are now working with the clinicians interested in working with us to see if we can actually develop a collaborative project. So this work, I'm kind of summarizing 3-4 years of work in a single slide here, which soon will be published. So that brings to the other fun part. I think I have some time to go through it. So the ecology of Powassan virus transmission. This work was started after I moved from Texas to here. Unfortunately, because in Texas, there is no Powassan virus. So I couldn't do anything there. So as we moved here, I wanted to do that. And the primary reason is this, because we know that Powassan virus is emerging and the Powassan virus is highly focal in its emergence. We don't know what it is, right? So, but the factors could be many. Non-traditional vectors are there, climate change, landscape habitat changes, human behavior, that's anthropogenic influence, and viral genetic divergence. Obviously, we all know from SARS-CoV-2, right? Keep on mutating, the spike protein keep on changing. The same thing can happen for Powassan DT, right? So you want to kind of understand that. So what we did, like any other epidemiologists and ecology people, we decided, okay, we moved. So we came March 1 of 2019. We decided, okay, let's go to the field and get the ticks. We thought that, okay, we're gonna get, so New York is an epidemic area. Let's go. We will, right away we'll get our positive ticks. We were in for a surprise. No. So site of the New York State, every state county required me to get a permit. And there's only like 0.1 to 4% data ticks are positive, which means that it's like searching a needle in a haystack. So that kind of put us in the back burner. But however, it allowed us to develop a lot of tools and techniques. So when I moved from Houston to Syracuse, my entire lab moved with me, and I need to give them some work because our biocontainment lab was still under construction. So I can't do my Powassan laboratory research because it's a BSL-3 virus. So we decided, okay, that's a perfect time. Let's bring everybody into the field. So that was my dream team that came with me. And those 5 people that there, they made my lab successful, what we are today. So those 5 people helped me develop this program. That is a passive surveillance program. So we decided that, okay, we are having all the problems to go into the state. So we decided, okay, let's sit back, reverse the strategy. We allow the people to send the tick to us, and then we test it, and then we will know where the virus positive ticks are there. That was a very simple one. Originally, it was started for Central New York. We didn't know what our capacity is, but due to the overwhelming public support, we went to Upstate. Within 2 weeks, I decided, okay, I got to open up to the entire New York State. So that's why we did it. So a brief overview, I think I have some time to go over it. So when a public find a tick, they go to our website, complete some form, submit the tick is received in the lab, we surface sterilize the ticks, we then morphologically identify under microscope, we then collect a lot of other data in the lab. So we actually, the first public submission form is provided by the public. The other one is what we collect in the lab. So we collect about 46 sets of data for every individual tick that is sent to the lab. We then grind them. We then do PCR analysis on them. We have developed our own multiplex qRT-PCR panel that detects 16 different tick-borne agents that are most commonly present in the northeastern United States. In addition to that, we only use half of the samples for our tick identification, pathogen identification. The other half actually goes to our freezer. And you will know why, because sometimes when we find interesting virus or pathogen, we want to culture it, we want to investigate it. And that's one reason why we don't want to waste our tick samples that we're receiving. So we now have a biobank archive, a freezer full of our samples that we can actually provide anybody interested. I'm a virologist, so I like to do every virus work. If anybody interested in Borrelia or Anaplasma or anything, we are more than happy to share with them. So as soon as the ticks are tested, it goes into our database. As soon as it goes into the database, we have a couple of gatekeepers to verify the results. As soon as we verify, we send an automatic email to the submitter, and also the data is used to create our own dashboard that anyone interested can go and look at the nyticks.org. So here is a list of pathogens we are testing. But again, I must tell you that originally our intention is to test only Powassan virus. But then because the overwhelming support and the opportunity and the gap, because nobody was investigating in the granular way that we have been doing. So I thought that, well, I have to exploit this opportunity to do everything rather than just Powassan virus. That's how we decided that to increase our panel now, we do it. We also look for Tick-Borne encephalitis virus. We know it's going to be negative, but it was more of an academic interest for us. And this is here is a list of everything that we've been detecting. And I must tell that the Babesia duncani is something that we have been detecting in ticks That is something that we are working behind the scenes to make sure that everything is right. And I think that is something that there is no prior documentation of Babesia duncani in the New York State. So we have found it. We are working behind the scenes to make sure everything is correct before we can publish that information. So here is the dashboard. Anyone can look. It's available to public. Easy to retrieve data. Date range option is there. You can actually scroll over the mouse. You will find everything. However, this talk is not for this, so I'm not going to go into the detail. But feel free to go to nyticks.org or tickmap.org, or our lab website. You'll be able to do it. It is available. However, it's not growing because we kind of put a pause to the program because we don't have funds. I don't have money to it. So here is a kind of a map to show that we have tested almost 27,000 tick human encounters. I don't call it ticks anymore because ticks are those things that I go to the field and collect the ticks, right? But these are encountered by human. Every single tick that was sent to my lab is encountered by someone, either on their pet or on themselves. So I call it tick human encounters. So that is a differentiation between active surveillance and passive surveillance. And as you can see from this map, we have tested ticks or received ticks from everywhere in the New York State. I don't show the map of United States because I want to kind of hone in on the Powassan virus soon. So we collected all these ticks, although we have all the data, but our original interest is Powassan virus. So that's where I narrowed down. Okay, now we know 0.34% of the ticks that are sent to the lab, they carry Powassan virus. Amongst them, deer ticks are the majority, followed by a couple of Exodus cookii. We have received females that are positive, larvae, adults, and all stages of them positive. And you can see this is a kind of a overview over the years. The first tick that we got was in July of 2019, and we continue to receive positive ticks until 2022. I don't have the data for the 2023 because our program kind of put on pause from March 1, but you can see clearly that together, if you kind of recall the epidemiology data, over the years, the tick human incident case was on the rise. We saw a similar pattern in our tick testing as well. So these are the ticks humans are encountering. And we kind of saw a correlation between what happens in the humans and what happens in the ticks. So there is a direct correlation here. So amongst those ticks that are sent to us that are positive for Powassan virus, 72% came from humans or encountered by a human. The 18% was attached to a dog, 6.9% to other mammals, and 3.4% on a horse. And if I can actually draw your attention to the other one, 75% of the ticks that are positive for Powassan virus that are sent to a lab are partially engorged, which means that they have spent a good amount of time on the host taking the blood, which means that they have enough time to transmit the virus back to the host as well. Only 11 of them are unfed, which means that they may have been crawling on them, or when they were doing the tick check, they pulled it. So we didn't detect any sign of tick feeding or blood feeding, so we call it unfed. So we allowed this data or tick mapping data to hone on identifying the hotspots. So that is our original agenda, is where these viruses are actually emerging, right? So we were able to, with this, our 3 years of data, we were able to identify the hotspots. So now we are doing habitat ecology, climate change analysis, and viral genetic divergence. It's a lot of things that we are trying to do. We're ambitious, but I know we are not going anywhere with that. Again, funding is very imminent for these kind of studies. It's a longitudinal long-term studies, so we are not going anywhere with that. However, so this is a kind of a experimental plan, exactly what we are doing. So we want to do a holistic One Health surveillance. We have hotspots, and we kind of divide a hotspot in different zones where we do in-depth surveillance analysis for vectors, habitat ecology, and human surveillance as well. We haven't started the human surveillance, which is something that I'll be starting it very soon. And we place camera traps and weather monitoring systems there. And you can see a camera on the right side here and a mouse that we are collecting. That mouse has no ticks on them. And then we do tick collection, active surveillance as well in our field. But what we are doing beyond this, so all these things are in the field, right? And then we want to combine our laboratory study into the mix because I'm a laboratory scientist to start with. So the viruses that we have been collecting from the field and the citizen science, we want to characterize them. We want to characterize them to see, primarily our primary objective was genetic divergence. So here's an experimental overview of how we do it. I'm not going to give you a lot of details to burden you. So we have received ticks through the citizen science program. That's the engorged tick. The ticks collected from us from the field. We grind them individually, and then we take the homogenate, intracranially inoculate the mouse. And then when the mice get sick or not, by 10 days, we harvest the brain. We then homogenize the brain again with the assumption that the brain will have virus in them. And then we culture them in a small petri dish. And then we sequence them, and then characterize them by re-inoculating in the mice to understand the pathogenesis. Are they virulent or not? Why people are not getting sick? If there are so many ticks that are positive, why people are not getting sick? So we want to understand that. So I want to give you some example. So here are the three virus that we have isolated from human submitted ticks. On the right, 7771, it is from a human from Orange County, encountered on 2020, November 2020. It's a partially fed female, and it was co-infected a female with Babesia microti. As you can see here that, so CPE is actually morphological changes due to virus infection. When virus infect a cell, they burst out, they kill it, they become more, they go to the next cell, they burst out. So when it does, it actually kills a cell and forms a small empty space where there are no more cells there. So the gaps that you see on the left are cytopathic effect. When cytopathic effect happens, that we know that virus is replicating happily, robustly. So on the left is an example of a high CPE. On the middle one is a low CPE. So you can see that there are healthy cells there. On the right one is low to no CPE. The middle one is an unfed tick from Columbia County. That's our hotspot. And the right one is a human. That one, I have a slide to show. It is from Westchester County, encountered on 2021, partially fed female, co-infected microti. So you can see that on the left and the right one, both are co-infected Babesia microti. However, one was highly virulent, the other one is less virulent. So we are now putting this virus isolate in a mouse model to see what really happens. And also we are going to sequence the full genome of this virus to know, are there any viral determinants that actually makes this one, replicating faster, the other one replicating slower. And this could be the reason that maybe this virus would actually cause the person to have meningitis and encephalitis. This will be asymptomatic. So that's our hypothesis. We had to test it out. And also we wanted to see the foci morphology. So foci morphology is basically when a virus gets into a cell, it actually replicates. So we have a way to detect this replication event by immunogenically staining the cells. So on the left is our prototypical strain. That's the original DFT virus that was identified, isolated by the Harvard researchers at Wisconsin, Connecticut. That's the strain that we use for all of our research. On the right side is the one that we collected through our citizen science program. And you can see that they all come with different sizes. The 771 that I saw, highly virulent, that was killing the cells faster, you can see it has a larger size, as good as the original one, which means that that is probably an actively replicating virus. And you can see the one that we didn't see any CPE, smaller dots, which means that the virus is replicating very slowly. So there is something that is stopping the virus from replicating faster. So we don't know that yet, but we're going to investigate that further. So this is a kind of a superimposition of the slide to show what we observe and how the virus is replicating versus how they are killing the cells. It's more of a comparative thing. But what I want to kind of hone here is that this is a greater example of telling that there is no one strain of virus circulating in nature. There are multiple strains with differential virulence capacities circulating here. So this tick submitted 2 ticks to us. The ticks were actually found behind her neck. They sent it to us. And then as soon as my lab detected positive for that, I know it was nearly fully fed. So I emailed the person and say, hey, by the way, you will get the results tonight. However, I want to reach out to you earlier that your ticks are positive and this was fed on a human and partially fed. So here are this clinical signs you had to look for. If you have any question, call me. 5 minutes after, she called my office right away and said, yes, I was sick, but I'm recovering. She went to the doctor. Doctor said, hey, go and get Tylenol over the counter. She went back, she recovered. So the long question short is, this is an asymptomatic infection. She got recovered, but she was never diagnosed with Powassan virus. So this is a wasted or missed opportunity for me to follow through with the patient because we were so busy testing the ticks, I couldn't follow through it. But later I went immediately the same weekend. I took my entire family for tick collection because I was so stoked that, okay, this tick fed on somebody was sick, that property must be full of positive ticks. So I went there, we collected the ticks. Yes, exactly. There's a lot of ticks that are positive. So now it is one of our hotspots. However, it is a long drive for us. So we are not, that is a little bit, the rest of the hotspot for us are around Albany. This one is Westchester, so I'm a little bit not sure if this is a good idea for us to keep traveling that far. So we keep it this site as a backup site. But this is an example I want to share that not all viruses make the person sick. Some febrile, some meningitis, some meningoencephalitis, some succumb to the disease. So all of them are circulating in nature. So the major outcomes from this ecology studies, we now have 73 new isolates from ticks. No one else has it. And I can proudly claim that this is probably the maximum number of virus isolates any academic lab has, even more than CDC. And of that, 10 are Powassan virus, lineage 1, 63 are deer tick virus. So we're gonna do our experimental design on all of these things in the future, and also try to understand why there is a genetic divergence happens, and is it correlated to a geographic location? Is it correlated to mammals that are present there, or is anything else that is actually influencing that differential virulence? So multiple variants of Powassan with deer tick virus with different virulence are in circulation. So on that note, I'd like to thank my lab people, also our collaborators who do extensive gene sequencing, virus genome sequencing, generous support from our funders, and also the community partners, Upstate Foundation and CNY Lyme Alliance, and my academic partners, the Global Health and SUNY Center for Vector-Borne Disease. So with that, I'll be happy to answer any question you may have.
One of those handhelds, thank you.
Oh.
Doctor. Sorry. No, not at all. Put this one over there. Please identify yourself for the Zoom audience.
Robert Lockhead, Medical College of Wisconsin. I really, first of all, was upset that Wisconsin was mentioned so much at this talk. And I really appreciate your work. More seriously, I mean, arborviruses are rare until they're not. And we've learned that. I have a question actually about some of the beginning of your talk. Males, younger males, more susceptible, and you kind of said, well, because we don't check for ticks. I'm not so sure. I want to challenge that statement. I wonder if, Dr. Laydet, my comrade here, said that different mammals have different competencies. I wonder if that's the case for humans, because Lyme disease has a very similar younger male prevalence. I'm wondering about that. If you could comment.
Question. The male reference was mostly a comical relief from my boring talk. But however, I must tell that in laboratory pathogenesis studies on mice, we don't see any difference between male and female. So the study that I showed you, a one slide neuropathogenesis study, we did both on males. So we did 10 males, 10 females, all looked the same. We couldn't find any big difference. We did even next-gen sequencing on the brain of those mice. No, we couldn't find any big difference. The only thing is the male-specific genes were upregulated, that's it. So it actually allowed us to conclude that at least in a small mammal system, in the animal model system that we use, there is no difference between males and females. But we see a change here, but we don't know it. So hopefully when I have access to the brain samples from clinically dead people, maybe we will try to understand a little bit more if there is any genetic bias or what determines the genetic bias. So that was a comical relief. It was not a scientific information.
Okay, next question. Please identify yourself.
Hi, we have a couple of questions here on the Zoom. I'm Keith Wilson. I'm just a moderator. The first one is from, I don't have the full name, but McGrath. Have any antivirals been tried for Powassan virus?
We have tried in the lab, so it works in the lab, but I don't think there is any official prescription to use antiviral for Powassan virus infection. No.
Thank you. Thank you. And then the second question is, how do you know your graphs of tick bite increase are accurate? Because you're using citizen science, is it possible that the increase in ticks that have been sent in could be attributed to more people hearing about your program over time?
That is highly, highly possible. It is highly possible. So that is something that we are trying to ascertain. So we are working with public health staffs to see if there is a correlation between our popularity versus really emerging. That's something that we are working on it. However, I must tell that we somehow reached a plateau between 2021 and 2022. The number of ticks we received was approximately around 10,000 ticks each year. But however, the Powassan number of ticks actually started to grow. In fact, 2021 was our highest year. So there is an increase. So within our data itself, we can tell there is an increase, but that is something that we acknowledge it. It is possible that we are getting popular, but we don't know that until we do a statistical analysis on that. That's something that we are working with our Upstate Public Health to dissect our data.
Hi, Monica Embers, Tulane University. In Europe, there is a vaccine available for tick-borne encephalitis virus. Do you know, is that vaccine cross-protective against these strains? I know they're similar, but they're not completely similar based on your sequencing. And or could you use those principles that are applied to that vaccine to develop one for Powassan, given that it appears to be expanding?
Yes, the TB vaccine that is given widely in the Europe is now available in the United States as a travel, vaccine. You can go to any travel clinic now, get it. FDA has approved it, I think last year. That provides 50% cross protection to Powassan virus. But again, that's done through in vitro studies, but I wouldn't recommend that, to be honest, because we are starting to see more adverse reaction. If someone gets a Tick-Borne encephalitis vaccine and they get exposed to Powassan virus, I think there is some information evolving that you may be actually exacerbating the situation rather than actually helping. So my lab used to give vaccine to everybody. Everybody who want to touch the Powassan virus vial, I send them to travel clinic to get it. Before FDA approved it, I send them to Vancouver or send them to Germany to get the vaccine. But now I told people, no, because now I see a little bit more scientific information, it may not be true anymore. But the literature says 30% cross protection.
Other questions, raise your hand. Go ahead on that side, Chris.
Chris Plano, University, Upstate University. We've had these conversations before. Like, I've always been familiar with the other flaviviruses that we work on here at Upstate. And when I was in the military, dengue coming to mind. And you already kind of highlighted the fact that there's probably a tip of the iceberg kind of situation where there's only the most severely affected patients that are showing up and being tested. There's potentially, like, you're the patient that you mentioned that, you know, either they're minimally symptomatic or maybe a lot of asymptomatic patients. And I know, I think it was Michigan, they did that seroprevalence study, maybe Michigan or maybe Wisconsin. I can't remember which it was, but that's been done in other states in asymptomatic patients. Have you ever thought about doing that in New York State? I'm not aware of anything that's ever been done. And it would be something, at least from Upstate, you know, we cover 10% of the state. Everybody that comes into the hospital gets blood drawn. That's basically just kind of held for 5 days.
I think Upstate Global Health is doing it or want to do it. And that is something that we want to do it as well in our hotspot area to actually do human serosurveillance. And as we speak, I think Michelle is actually giving us some options where we can actually piggyback on your protocol to kind of do this surveillance. Initially, our idea is to go to the hotspots and do it. And also to those who send in the ticks that come as positive, we want to reach out right away to them. Okay, I want to put you to like a sample collection protocol so we can monitor them. So we do have a little bit more clear, defined objectives to do the serosurveillance. We don't want to do blindly everywhere because that will burn me down already, because we are running the lab frugally. So I want to kind of invest our resources wisely, but to go to the hotspot to do the human surveillance and also to those people who send the tick to us that are positive, I think those are the primary things that I want to approach it, and we are working on it. I'm hoping that we should be able to kickstart that as soon as possible, maybe in the next couple of months. But it's going to be your protocol.
Questions, raise a hand. You got a mic? And way in the back. Yep.
Hello, Christine Herb from Syracuse. And is your lab going to be testing ticks in 2024?
Well, that's the million-dollar question. I want to start. There's a lot of things going on behind the scenes that indicates that I might be starting, but there's a lot of discussion going on. But I don't know. I don't want to say anything officially yet, but it is my wish to start the program again, because I personally know how our program was helping people. Because like you said, paying $150 for testing, we were doing for free. So I want to do it, but there's a lot of caveats and ifs and buts there, but we are actively working behind the scenes to make everything possible. Us and Upstate is trying to get it done. And I know that the county executive that gave a talk, he opened up the funding for us to start with. Hopefully other counties will follow that as well. But soon we'll announce, probably around November, I'll make an announcement.
Stay tuned. Other questions before we break? We have an opportunity for a break, but not until all of your questions for Dr. Thangamani have been addressed. Anybody else? Put a hand up and we'll come to you. And if not, it's coffee break time, but no later than 11:10. We'd like you back in this room to
Thank you
start
for
again.
attention.